Okay, so welcome along, ladies and gentlemen, to your weekly SETI seminar series. Today we're very lucky to be joined by Tori Hoehler, who's come across to us from NASA Ames, where he's a research scientist in the exobiology branch. Tori got his BS in chemistry at University of North Carolina and then did a PhD in marine chemistry and biochemistry from the same institution. And then he came across to be an NRC fellow at NASA Ames. And then since then has joined the staff at NASA Ames. His interests are in microbial ecology and biogeochemistry, hydrogen cycling, subsurface and low energy ecosystems, and in planetary habitability. He's published in areas such as environmental constraints on methanogenesis, microbial mats and production of reduced gases in the early Earth's atmosphere. He's published on energy balance concept of habitability and the ongoing mystery of seafloor methane. And so today he's going to talk to us about carbonates and serpentinization. So please join me in welcoming Tori.
Thank you. Can you all hear me okay? Thanks for the invite. Happy Guy Fawkes Day, which interestingly enough falls on Election Day this year. Yeah, well, I'm not implying anything at all about. I'm not implying anything about the coincidence of those two things in this particular year. But and thanks for letting me give you a snapshot of work that is in progress. So this is not a finished story by any means. It's a project that we have ongoing, not very far north of here in Lake County, actually. And it's been an opportunity to have a look at the process of serpentinization and how it influences life. And I'll try in this talk to go over a few things. So I'll very briefly talk about what serpentinization is. It's become quite a popular topic lately, and I think more processes are called serpentinization than in reality actually are serpentinization. It's become kind of a catch-all term to describe any reaction of water and rocks, and that's not universally true. So I'll talk a little bit about that. I'll talk about what I think all the fuss is about when it comes to serpentinization, and in particular, why this is a subject for a colloquium in relation to space, or why the folks who pay my paycheck care that I work on this project. I'll talk about the current limits, I think, where we are in this field right now, and at that point I'll pull up a soapbox and talk a little bit about my views on habitability and how we currently apply that concept, some modeling work related to this process of serpentinization, and then the project that we have to actually look at the microbe community associated with serpentinization. So here is what the process of serpentinization is, and very briefly, and I'll revisit this, serpentinization is the reaction of rocks very much like the rocks that our own mantle is made out of with water, and it makes this mineral, which is serpentine, which gives the process its name. You can see serpentine all over the place here in road cuts. If you see the sort of pale green color in the road cuts on 280, that's all serpentine. It makes some alkaline minerals. This one is brucite. And what gets people really excited is that it also makes hydrogen. And that gets people excited because hydrogen happens to be good food for microorganisms, a lot of microorganisms. And so people have viewed this potentially as a way of supporting a microbial community in the absence of sunlight, let's say. So this is a process that could actually work down in the subsurface, even when you have no access to the surface world. And here's an example that people often like to look at is the process of methanogenesis, where really all you need is some carbon dioxide, the hydrogen that you might get from this process of serpentinization, and there are microorganisms that can make a living by catalyzing that reaction. And this is the reaction that actually I'll use as the example for the remainder of the talk. So there are a couple of observations about this process over the last decade or so that I think have served to get people excited about it. And the first was the discovery of the Lost City hydrothermal field a little more than a decade ago. So this is just off the mid-Atlantic ridge, and in contrast to most of the hydrothermal vents that are known, that are hosted in basaltic rocks, come out at something like 300, 350 degrees associated with sulfide minerals, this one is different because it runs on this process of serpentinization. And what got people excited about this is that there was a very diverse and abundant biology associated with this field. And so it gave support to the notion that maybe this process that's generating hydrogen actually could be a viable habitat for life or a viable basis for life. And as well, more recently, there's been a detection on Mars of both the starting material, olivine, and the product material, serpentine, of that reaction. So the process has been happening on Mars. And one of the things that's significant about this is the rocks, as I said, that comprise our mantle, that are the basis for serpentinization, are also what you expect to find on Mars, in the subsurface of Europa, for example. And so there's potential that this is something not only going on here, but elsewhere. And so this detection of serpentine and the mineral assemblages associated with serpentine, actually in quite a few places across the surface of Mars, are maybe giving some support to the fact or the idea that this is a pretty widespread process. And this got people excited about the idea that, well, you know, if this is happening on Mars, perhaps this is a viable habitat for life there, maybe even in the present day if the process is ongoing in the subsurface. But I think this is, it's sort of important to think about how we apply the notion of habitability, because that's what we're talking about here. You know, could the subsurface of Mars in the present day or could Mars in the past have been habitable as a result of this process ongoing? And the way that we view habitability, I think, at least in the way that we apply it in planetary science, is to say, well, you know, we've collected some evidence that suggests we have found an environment of a certain type, and on Earth we know that such environments are capable of supporting life. That's about as far as we've gone with habitability. In reality, if we look at the world around us, habitability, if we take a deeper view of it, is really understanding how life distributes itself against a landscape of physics and chemistry and geology. And if we look at the example of the world around us and we take as the parameter with which to quantify habitability biomass density, so that's one among several different parameters that could reasonably be used to talk about habitability. If we take the parameter of biomass density and we look, for example, at the continent of Africa, you can see a variation of maybe 5 orders of magnitude or so in the amount of biomass that is supported from the equatorial regions up to the Saharan region. And that distribution, as we know, is largely underlain by the presence of water. It rains a lot here, it doesn't rain very much here. But the point is that life distributes itself in a continuum. It's not 1 or 0. It's not none here and some here. It's a continuum of 5 orders of magnitude. There's very little here. This is very vibrantly inhabited. And that pattern holds up regardless of which variable we look at. So if we go to a place where there's water all the time, so if we look at the surface oceans and we quantify the amount of chlorophyll, that's a proxy for the amount of photosynthetic biomass in the surface oceans. So here the hot colors down to the cool colors represents about 3 orders of magnitude range in how much biomass. So the same parameter, water is ever present in this environment, and yet we still see about 4 orders of magnitude in variation, again in continuum from one place to another in the ocean. And this is largely underlain by the availability of nutrients, so it's a different parameter than water. And if we went to these sites out here, and we looked in the sediments down at the bottom of the ocean, so this is a place where the sun don't shine, water is always present. In these places, we find a distribution, and this is the density of cells per milliliter as a function of depth in the sediment column. Across a whole variety of sites, we see something like 8 orders of magnitude variation in how much biomass there is here to how much biomass there is here, and that distribution is completely underlain by the availability of energy. And I would argue that this is actually the most relevant one to think about when we start thinking about, is life possible in the subsurface of Mars or Europa or so forth. And there are a couple of points to make about this. The first is, habitability is not a 1 or 0 proposition. A place isn't simply habitable or not habitable. Life distributes itself in continuum across many orders of magnitude. And there's a richness of information there that really isn't captured by the way we currently apply the notion of habitability. But that's possible for us, or at least progress down that road can be made. And the other is, habitability is about more than just water. And in fact, once you've checked the box of water, the parameter that probably matters the most for consideration of Mars or Europa is the availability of energy. And I'm going to try to take a look at the habitability of serpentinizing systems through that lens of how energy influences habitability. So let's come back to this process of serpentinization. And I represented it in this way before. You often see it represented in this way in textbooks. But the point is, you know, this is a notional scheme for how this process works. And in fact, there can be a lot of variation in how much magnesium or how much iron is in there. The other conditions in the reaction can influence what these products are and whether hydrogen is in fact made or how much it's made. And so really to encompass the entire thing with one single reaction really fails to capture the richness of these systems. And if we look back again at this place, Lost City, the features that we most associate with serpentinization pop out at us. So this place, Lost City, the fluids come out with hydrogen concentrations or activities up to about 15 millimolar. And I know that that number doesn't necessarily mean anything. For my dissertation work, I studied hydrogen cycling in marine environments. And hydrogen there, which serves as an ample basis for the biology, has concentrations about 6 orders of magnitude lower. So this is a screaming high hydrogen concentration. It would be great for anything that lives there. And that's the plus side of serpentinization. The downside is very often it's associated with high temperatures that make the serpentinization reaction go faster, and very characteristically, very alkaline fluids. So these are natural fluids that at the Lost City have a pH up to about 11. So household ammonia is pH of about 11.6 or so. This is a very alkaline environment, and that's really challenging for life in ways that I'll describe later on. But again, this place has a lot of evidence for biology associated with this process of methanogenesis. And this has gotten people really excited. But the thing is, it's not just a single chemical reaction. And this is a set of plots from Tom McCollum a few years ago that shows that if you take the very same rock and the very same water and you simply vary the parameter of temperature, as he's done here, there's a real significant variation, first of all, in the stability of the minerals associated with this process. And as a result of it, there's a real variation in the pH as a function of temperature, and pH is a problem for those microbes, as well as in the production of hydrogen associated with this reaction. And so if you were to look at this in qualitative terms, you would say out here, where the hydrogen is abundant and the pH is relatively moderate, those are great conditions for life. Down here, where the pH is very alkaline and the hydrogen is very low, just in qualitative terms, that's not nearly as good a spot for life to live. And that's the sort of richness that we need to capture with our approach to understanding habitability in serpentinizing systems. Moreover, if you look at the type of rocks that are involved in this process, it's not simply that there's a type of rock that reacts to serpentine. The rock can have varied concentrations, for example, of calcium, as it does here. And calcium turns out to be a very big driver of the production of alkalinity in this system. So here, if you look across the panels, is very little calcium, a little bit more still, and even more. And this more or less captures sort of the naturally occurring range of calcium in the rocks that serpentinize. What you see represented in the colors here is pH, where very warm colors represent very high pH. So when there's relatively little calcium in the rocks, there's not very much potential for the system to get very alkaline. If you look up in a system that has a very high proportion of calcium, you actually can get pHs in the range of maybe 13, close to 14 or so. So there's one variable that matters, and that's a function of the kind of rocks that you're looking at. The amount of iron that's in the rocks also matters a lot. That's what generates the hydrogen in the process. And the simple point is, you can't just say that water reacts with rocks and it makes alkaline conditions with hydrogen. That's something that you can quantify as a function of the kind of rocks that you're looking at, and it's something that matters very much to the biology that might be associated with these places. And that's reflected. If you look at a handful of places where this process of serpentinization is actually influencing the chemistry of the waters, here are 3 of the vents associated with the Lost City hydrothermal field. Here are a couple of wells from just maybe 10 or 15 miles south of here in Santa Clara. Here are a couple of places sampled by former postdoc of mine, Don Cardes in New Zealand. And here are the parameters that would really matter to biology associated with those systems. In particular, it would really matter to methanogenic, methane-producing biology. Hydrogen, of course, as the food supply, and I've ordered these from the most hydrogen, the highest hydrogen concentration, down to the least. And I'll just draw your attention to the fact that in these systems, all of which are associated with serpentinization, there's nearly 5 orders of magnitude in variation between the most abundant and the least abundant hydrogen. pH, which also matters, Friedemann.
What is the solubility of hydrogen in the water? There's a limit. Hydrogen as a gas.
There is a limit. So at the question was, what's the solubility of hydrogen gas in water? Here at atmospheric pressure, it's about 1 millimolar. So it would be just about here. This is under about 800 meters of water, so you can actually cram a lot more hydrogen than that, which allows it to get higher. But the simple point of this slide is to say in each of the parameters that matter, so in hydrogen, in pH, in dissolved inorganic carbon, so you can think of that as CO2, there's very significant variation. that should underlie a real significant difference in what the biology feels and what is the potential to support life. So that's something that we should be able to capture with our concept of habitability. And there's good, bad, and ugly to this if you want to look at the effect on biology. So the great thing about serpentinization, if you're an organism that catalyzes this process of methanogenesis and you like hydrogen, that's the place to be, because this process has the potential, at least, to generate a lot of hydrogen. So the good thing about serpentinizing systems is the hydrogen they generate. The bad thing is, very often, that's part and parcel to a lot of heat. So very often, these systems have some heat underlying the circulation. At very least, Lost City does. And that's a problem. Heat is more difficult to deal with if you're biology. The pH also is high. That's a problem for biology. But the really ugly thing, if you're an organism that depends on carbon dioxide for your metabolism, and you live at a pH of 11 or 12, you have a real issue, because there is no CO2 at those pHs. Almost all of it is speciated into bicarbonate or rocks made of carbonate. Okay, so there's a real problem of substrate availability. And almost always on Earth, people think about this process as depending on hydrogen. But in these particular circumstances, this carbon dioxide and the availability of carbon dioxide become a real issue. So those are quantitative statements, right? Some hydrogen is good, some alkalinity is bad, a problem of CO2 limitation is bad. Those are qualitative statements that should be quantified in our concept of habitability. And here's an attempt to do so. So I'm going to think of these in energetic terms, and I'm going to try to quantify them in reference to how they affect the capability of an organism either to take in energy or the cost in energetic terms of living in an environment with those characteristics. And an easy way to think about the requirements of biology for energy is that it kind of breaks down in the very same way that this laptop has a requirement for energy. If I could flip it over, I would see that it probably takes something like 19.5 volts and maybe 100 watts, let's say, right? So that's a requirement for voltage, and it's a requirement for power or energy flux. And you can think of biology in exactly the same way. We have a requirement for voltage, which in chemical terms we would describe as a Gibbs energy change requirement, and as well for power, right? I need a certain amount of power per day. And the power requirement is easy for us to equate to everyday life. Okay, I eat a diet of, let's say, 2,000 calories per day. That's power, right? Those calories per day, that's units of power. And I maintain an approximately constant size. So it takes at least that much energy to keep me as the standing version of me. And I've been eating a diet of something like that since I was 18 or so. I've weighed about 165 since I was 18 or so. I've consumed something like 10 gigajoules of energy in order to keep me exactly the same size that I am now, and as a arguably functioning individual. Okay? So all life is like that. All life has those requirements for voltage and power. And those are handles that we can place on the environment. It's a way that we can look at the chemistry, because we can understand from the chemistry and the physics of diffusion how much energy is available, how fast it reaches the organism, and we can compare it against what the organism needs to keep alive. So let's have a look again at these same fluids with the diversity here in hydrogen and pH and dissolved inorganic carbon and so forth. And almost always when you ask a chemist or a geochemist to talk about energy availability in a system, this is the answer you'll get. There's a calculation of Gibbs energy availability for this process of CO2 plus hydrogen leads to methane. And any number that's negative means the reaction is favorable in chemical terms. Any number that's more negative than about minus 20 or so means that the Gibbs energy requirement of the organism is met. When you look at these, there's only one of them down here that fails to meet the Gibbs energy requirement, and even that one is arguable. I think if we begin to look in natural environments, the Gibbs energy requirement maybe is closer to minus 10 or so. So these fail to resolve from one another because the Gibbs energy condition really is an if-then requirement. If the energy available exceeds something like minus 20, life is possible. If it doesn't, life is not possible. So all of these places, despite the significant differences in hydrogen and pH and CO2, fail to resolve from one another by this metric that really is the standard way of looking at energy availability in an ecosystem. And actually, that's not terribly surprising. And I'll give you an example of why. It's because if a reaction occurs fairly far from equilibrium, that means if the Gibbs energy change is relatively large, it's difficult to make it change very much by changing the environmental conditions, so by changing the concentrations or the temperature or so forth. And here's an example that is only slightly silly. So let's look at this same process of methanogenesis. Here's CO2, and hydrogen goes to methane and water. And we can calculate a Gibbs energy change associated with that reaction. So the conditions that I've applied are a hydrogen concentration of something very close to what you see at the Lost City hydrothermal field, similarly with the methane concentration. And then what I've done is varied the CO2 concentration and calculated the Gibbs energy yield as a function of that concentration. So if we start up here at a CO2 concentration of about 10 to the minus 3, which is kind of what organisms would see if you grew them in pure culture under conditions that they really like, you get a nice fat energy yield of about minus 150. And then I've dropped the CO2 availability in steps of 6 orders of magnitude. So there's 1,000,000 times less here, 1,000,000 times less here. And you see that the Gibbs energy change gets less and less negative. That means less favorable. But you have to drop by something like 23 orders of magnitude before you get to a place where it's even questionable whether life can make a living. And you have to drop down to a concentration of about 10 to the minus 30 before the process ceases to be thermodynamically unfavorable. And we throw around big numbers all the time in astronomy or chemistry and even biology. Just to put this in realistic terms, a concentration of 10 to the minus 30 for CO2 is about the same as a molecule of CO2 in an Olympic-sized swimming pool worth of water. Okay, so it's kind of a nonsensically low concentration. And I use it to make the point that this Gibbs energy look at the environment has relatively poor resolving power when it comes to thinking about the energy requirements of life. If, on the other hand, I look for the requirement for energy flux from the organism, the equivalent to my 2,000 calories a day, and I use the exact same concentration, so the conditions are the same, the hydrogen is the same, I'm dropping CO2 by those same 6 orders of magnitude, what I see in terms of the power flux, which I've listed here in zeptowatts, because it's fun to say zeptowatts, what you see is that the power availability drops in steps of nearly 6 orders of magnitude. So life is very much better here than it is here. And once you get to this point, this is in power terms kind of nonsensically low. And the point is, this is a much better way, a much more discriminating way to resolve among environments in terms of how energy supports the biology. And that's a notion that we can apply to serpentinizing systems. What this lets you do is a couple of things. If you have a good way to calculate the energy flux, the metabolic energy production in an organism, and you also have a notion of how much energy it requires. So, I require 2,000 calories a day, and the environment around me provides 10,000 calories a day. I can do 2 things. I can divide one by the other. I can say, here's how much energy is available, and here's how much energy each organism requires. In the example I just gave, the environment could support 5 of me. Okay? The other thing I can do is subtract how much energy is available from subtract how much energy the organism requires from how much is available, right? And anything that's left over is available for growth. So I could use that as a notion of how fast can the system grow. So I've said 2 things about the physiological or the biological potential of that environment by quantifying energy available and energy required. And here's an example of how these things relate to serpentinizing systems. So here in this plot, I've done power generation divided by power demand. So that's kind of a notion of how much biomass could the system support. And I've done it as a function of hydrogen availability on this axis, so much more hydrogen available here than here by about 1,000,000-fold. And here as a function of pH, the 2 parameters that most influence biological potential in this system. And what you can see is that habitability isn't just 1 or 0. Habitability occupies a continuum of several orders of magnitude where the hydrogen is highest and the pH is the most moderate. Biological potential is very high. By nearly 1,000 times more than here. And importantly, as you run across this range of physical and chemical conditions, you reach a point where the system is no longer habitable in energetic terms. So energy is available there, water is available there, nutrients could be available there, all the boxes of habitability are checked. But when you start to be quantitative about it, you find that there are places in the system, places in the chemical space that are uninhabitable. And you could make a very similar statement in this panel about the potential rate of growth in the system. So if you do that, and you go back and look at, I've sub-selected a few of the systems that I showed you earlier. So here's one Lost City hydrothermal field vent, here's a second vent, and here's one of these places in New Zealand that Dawn sampled. If you looked at these, as most people do, just from the standpoint of hydrogen, you would say, this and this are really about the same, and this is not a very good place to live, because the hydrogen is down by 4 orders of magnitude. But these have different influences, and there are different factors to consider. The pH here is lower by quite a bit than it is here and here, so that's better. There's more carbon available here, so this issue of resource limitation is not as great here, and that's a good thing, right? So how do we quantify this? Here again, for reference, is the Gibbs energy yield, where the three systems really don't resolve very well from one another. If, on the other hand, we look at the capability to generate power metabolically, there's a great potential here, very little potential here, even though the hydrogen is very nearly the same, and that's because there's so little carbon available. That's the limiting factor in this system, and it's something that you can quantify with basic physics of diffusion. And sort of a modest capability to generate power here. But the cost of living in this environment is very low because the temperature is low, the pH is not terribly high, and it's not an awful place to live. So although the energy availability is lower, the cost of living there really isn't very high at all. And from the standpoint of how much energy it costs to live in this place, this generation divided by demand parameter, this is actually the best place to live. And you can say in quantitative terms, this environment could support nearly 5 times as much biomass as this one, and this one is, in this view, incapable of supporting biology at all. Okay? And the idea is that simply by quantifying this basic parameter, this basic requirement for energy flux, and using it to make statements about why hydrogen matters, why CO2 matters, why pH matters, you can resolve habitability across a spectrum, not just 1 or 0. You can do other things. You could take a walk across theoretical chemical space. So you could say, going back to this example of how much calcium is in the rocks, you could connect the notion about biology or the biochemical model to a model of geochemistry. And that's what we've done here. We've taken an off-the-shelf geochemical package, Geochemist Workbench, use it to predict how calcium influences pH in the system and how it influences hydrogen in the system. So here, this is low calcium, higher still, and highest. This axis is a function of how much iron is in the rocks. So here's significant amount of iron, very little iron. And what's plotted here again is this power generation over power demand parameter. And what you can see is there's a continuum across this chemical space. So habitability is very poor here. Habitability is best here at moderate water to rock ratios, at low calcium content, at high iron content. And what's significant about that is those are the sorts of things that you can hope to measure with spacecraft. Those are the sorts of parameters where you could look at the rocks and make predictions about what kind of biology they'll support. So that's theoretical, and it matters to see how this works in the real world. And so there have been a number of attempts to look at the chemistry associated with serpentinizing systems and a few attempts to associate the biological potential or look at the biology in those systems. The limits, I would say, though, and the current roadblocks to understanding are that this very wide variability in the physics and chemistry of these environments really is not well captured by the current sample set that we have. And in particular, almost everywhere that we've looked, we're looking at a surface expression of the process. So this is a process that happens deep with circulating fluids. People look at it where the fluids come out of a spring environment, and all of a sudden the chemistry changes dramatically because it encounters oxygen, it encounters carbon dioxide from the atmosphere. All kinds of things change all of a sudden. But that's the only window that we've had so far into these systems. And our understanding of habitability, could these places support life in the subsurface of Mars, is limited very much by that observational bias. Fortunately, there are many, many examples of these. So this is a map of the global distribution of known ophiolites. Ophiolites are places where bits of ocean crust have been scraped up by tectonic processes, lumped onto land, and the significant thing, freedom.
Crust. You meant mantle.
No, full sections of ocean crust. So the question was, I said crust. Did I mean mantle? The significance of ophiolites is that in this process of scraping up ocean crust, you're taking some superficial crust, maybe including sediments and pillow lavas and so forth, but also some of the underlying mantle and lumping it onto land. So you've taken some of these mantle rocks that underlie serpentinization, put them on land, and now they're accessible to you. Okay? You don't require a submarine to get there. All you require is a van. And that's something that we've done. So if you look at California, right, the fact that there's been a lot here going on geologically has left us with quite a number of ophiolites around here. There's an abundance of them, and that's a fact that we've taken advantage of by going to the McLaughlin Natural Reserve. So we are down here. The Bay Area is somewhere down south of this part of the slide. This is Lake County up here. Here's Clear Lake. and here's the McLaughlin Natural Reserve. And McLaughlin was a homestake or a barrack gold mining property, which ceased to become economically or ceased to be economically viable, and they thought the best thing they could do would be to turn it over to UC Davis and allow the establishment of a preserve. And it's actually a preserve for serpentine plant ecology, so serpentinization has a significant influence on vascular plants as well, very unique biota, and this is an observatory for studying that surface biology. But the neat thing about it is it's dedicated as an ecological preserve, access is controlled, there's an infrastructure for supporting researchers, and it's a perfect place for us to go and do the sorts of research we want to do. This is what it looks like on the surface. You can see here a little bit over in this part here. This is an outcrop of peridotite, so these are these sort of mantle-like rocks, and it's the circulation of fluid under these rocks that give rise to serpentinization. So the great thing about this being a former gold mining property is there's a requirement for remediation and monitoring. So Homestake has gone in and drilled a variety of wells down into this serpentinizing terrain. There are records dating back many years of what the chemistry looks like in those, and if you talk nice to them, you can go and have a look at them yourself, which we've done. So we did this several times running, and it allowed us to collect some baseline data about this place. And here is what we found. So here are a few of the parameters that I said are relevant to this process of methanogenesis. Here's the pH, and I've arrayed these as a function of depth. So these wells all sit within about 6 meters of one another. This one is about 1 kilometer away or so. But I've included it in this profile. So if you array them in terms of depth, here you get a pH of about 7.4, a lot of carbon in the system, very little methane. That's just essentially groundwater, so we think of this as a control. As soon as you go down just a few more meters, all of a sudden the pH is 11.5, a very clear signature of serpentinization. Very little carbon remaining available, tons of methane in there. But what's striking about these is here's the hydrogen in units of nanomolar. That's 1 million times less than is seen at Lost City. So already this place that is very characteristic of serpentinization is very different in chemical terms than Lost City, and therefore has a very different potential to support biology. And this is the sort of thing that's not captured well in our current sample set. If you go down a little bit deeper, the pH changes a little bit, more methane still, even more methane down here, but the hydrogen remains low. So what does that mean for the system? We can take a little bit deeper look, so we can look not only at CO2 in the system, but also at other forms of carbon. So here what you see is CO2. This is the methane in the system, which dominates the carbon in the system. We also looked at formate and at carbon monoxide. And the important thing about these is, number one, both of them can represent substrates for microbes, so each of them is potential food. But the other thing is you could look at each of them as being in equilibrium with CO2 and hydrogen. So you could write a reaction that says CO2 plus hydrogen goes to formate, or CO2 plus hydrogen goes to carbon monoxide, and it allows you to kind of do some forensics in the system, right? Because what we find is there's tons of methane around. So I wonder if the methane is a result of biology having created it or some other process that we don't know about. We can go back and ask questions, and we can address this concern or this mystery over why there's so little hydrogen in the system. So here's what I've done. Here's the observed hydrogen concentration in the system, about 12 nanomolar or so. And if you calculate, if you take the total amount of carbon in the system here, add up all of these boxes, and then you allow it to speciate or equilibrate at 12 nanomolar hydrogen, how much CO2 should there be, how much methane, how much formate, how much CO2? This is the answer you get. So according to this calculation, almost all of the carbon should be present as CO2, and very little as methane, and faintingly little as formate or as carbon monoxide. Okay? You can then ask the question, okay, well, clearly that's not the distribution that we observe. So what would be required to give us the distribution we observe? Okay, one thing that you could do in order to get higher concentrations of formate or carbon monoxide would be to have more hydrogen to start with. So maybe there was a lot of hydrogen from serpentinization. It all got chewed up and turned into methane, but perhaps there's some signature of that having been there. So you could go through and calculate and say, okay, in order to reproduce the distribution that I actually do observe, this amount of methane and a certain amount of formate and a certain amount of carbon monoxide, how much hydrogen would have to be there? So it would have to be at least 20 times higher to give me the amount of methane that I actually see in the system. And if I disallow that process from happening, so I just cut that part out of the model and say how much hydrogen is required to give me the formate that I see, it has to be another couple of orders of magnitude higher still. And to get the carbon monoxide that I observe, it has to be at about half a millimolar or so. So now that's kind of getting up into the lost city sort of range. And it makes us curious whether or not this was the reality of the system, whether there was a lot of hydrogen around and it all got chewed up and turned into methane. And we're simply seeing a residual. So this is it's a question that people are really interested in, and it really bears on the idea of, could these systems support biology? Because almost always you see methane associated with serpentinization. And nobody really knows. Was there a question?
Yeah. Can you use isotope chemistry on the methane to say something about what microbes produce it?
Yes, you can. The question was, can you use isotope chemistry? And I'll actually address that in this slide. So people are very interested in this question of where does the methane come from in these systems? Because if you can't find a viable abiotic mechanism, and you find methane associated with these systems all over the place, it suggests that biology is widely associated with these systems. Okay? So it's an important question to address, and it's one that we at least have potential to address here. So, there is a potential that the presence of methanogens in the system has given us the chemistry that we currently see. There's certainly a lot of methane, but as well, methanogens have an enzyme called formate hydrogen lyase that during the course of this reaction will equilibrate hydrogen, CO2, and formate. So there's a biochemistry that could potentially give you formate. And as well, there's a biochemistry that could potentially give you carbon monoxide in the system. So both of those things could conceivably be a result of methanogens operating in the system. So now we sought using geochemical means to understand whether that's in fact where it actually comes from. And the things that geochemists typically will do to identify or pin methane as being biological is, number one, to look at the ratio of methane to other hydrocarbons in the system. Biology is very, very selective and sensitive, whereas geochemistry is not. So if you see a ton of methane and very little of ethane and propane, usually that's an indicator that biology made the methane. The other thing that biology does is it's very selective with respect to isotopes. So if you have a molecule of C12 methane and a molecule of C13 methane, the biology will almost always pick the C12, okay? And it gives you a a methane product that's very enriched in C12 and very depleted in C13. So here are these two measures. Here's this ratio of methane to ethane plus propane, and it's about 3,000, which is a very biological looking signature. Okay, geochemistry or cracking of organic matter doesn't give you that kind of signature at all. The isotope, so typically what you would see associated with methane is the isotope for methane, which is represented here, would be more negative than the isotope associated with CO2 by maybe 30, 40, 50 or so, and there's almost no difference between them. So that really doesn't look like a biological signature. But the problem is those systematics for isotopes are almost never used in systems where carbon is limiting. And if you grab onto every molecule of carbon that you can get, regardless of whether it's C12 or C13, you can't afford to be choosy, and there's no isotope effect expressed. Right? So this is a system, simply because of the alkalinity in it, where you wouldn't expect any expression of an isotope effect at all. So we have a maybe indicator here, and a really ambiguous indicator here. Okay? But if we look at this reaction, CO2 plus hydrogen goes to methane, it's thermodynamically unfavorable under the conditions as we measure them in the modern day. So the depletion of hydrogen and the enrichment of methane that we see couldn't completely be accounted for just by this process of methanogenesis. On the other hand, methane can also be made from carbon monoxide, and that process is thermodynamically favorable. So we sought to have a quick look at that process. We actually did some experiments where we took these well waters. Some of them we filtered to remove any cells. Some of them we left intact. And we added hydrogen and carbon monoxide as the substrates associated with methanogenesis. And we left them for 2 weeks. So here are the controls, here and here. The blue is hydrogen. The green is carbon monoxide. And we compared the controls after 2 weeks, percent of original hydrogen and carbon monoxide remaining, versus several of these different treatments. Here's one that had hydrogen and carbon monoxide both. A second one that had the same thing, one that has hydrogen individually, one that has carbon monoxide individually. So the first thing that you see is there's definitely biology that is definitely consuming hydrogen and carbon monoxide in these systems at a pH of 11.5, right? As if it were working in household ammonia. So there's biology in here that's definitely doing something, but no indication in our experiments that it's methanogenic. So there are cells, but they're not methanogens. And this is actually consistent with what's been found in most other places that people have looked at serpentinization. Where you see consumption if you look at the underlying biology, almost never is it methanogens. It's almost always cells that look like they're seeing a little bit of oxygen based on their metabolism. And this goes back to this problem of always observing this process in the mixing zone, where fluids from down deep encounter oxidizing fluids from up high. And even in these home stake monitoring wells, that seems to be the pattern that we see. And that provided ample motivation for us to go and drill. And we got some director's discretionary support from the NASA Astrobiology Institute. We hauled a drilling truck up there and we drilled some holes in McLaughlin. And the purpose of doing this was it gave us subsurface access to fresh material. So we were looking at the process where it happens. These were dedicated wells that only we could use, and we could prescribe the way in which they were drilled. So we trucked in several thousand gallons of filtered drinking water every day to use as our drill fluid to make sure there was no contamination. We were able to strictly control the process. And we had resulting holes that are just about 3 hours or north of here, and that we can go and look at anytime we want. So we've established a microbial observatory where we can compare geology, because we have the core materials, geochemistry, because we can look at the fluid chemistry anytime we want, and biology. And that allows us to then make an inference and make a statement about how geology supports biology, so that when we don't have the last two, when there is no capability to measure geochemistry or biology, the geology alone allows us to make a statement about biological potential. So this is what it looked like from up close. We mostly drilled in mud, kind of became a joke, right? Every core that would come out, oh, let me guess, you know, it's serpentine mud, right? Kind of gray, pale green, whatever. We got meters and meters and meters of this stuff, but when it would come out, we would kind of dissect it and look, and we find a lot of the products of serpentinization. So the mud itself, the matrix, is mostly serpentine mineral with some magnetite in it. That means hydrogen was made in the process, with some carbonate in it, which means that CO2 was sequestered in that process. All the hallmarks of serpentinization. Here's what we found, and this is the work in progress part, so please bear in mind this is just a snapshot. So in terms of the bulk parameters, with this drilling and the subsequent monitoring, we've captured a pH range of something like 7, so a pure groundwater pH, all the way up to pH 12.7. One of the most alkaline fluids that has been measured in naturally occurring environments. And we have a chance to look at the chemistry and the biology across that entire range of conditions. Again, we see lots and lots of methane in the system. So in one of these places, when you pump out the water, it actually fizzes, and you can ignite the wellhead. But again, very little hydrogen and relatively little CO. So it's consistent with the chemistry that we've seen, only in a newly sampled, newly accessed environment. So part of the thing that's important for us, or interesting for us about this, is this range of pH that we've captured. If you look, as Billy Brazelton did in a publication last year, at diversity, sort of an arbitrary measure of biological microbial diversity in the system, as a function of the pH in the waters, you see what's maybe a not terribly surprising effect, that the diversity in the system, the kinds of biology that can be supported, drops precipitously as you get to higher and higher pH. And actually right here, that's our spot in Lake County, right? So here are all these different places that have been sampled. That's a pretty nice, robust, diverse community. As soon as you get to our Lake County site, there's really two things there. And really, really only one thing, as far as we can tell. So this is among the most alkaline fluids that have ever been seen. It has extremely low diversity. It's very difficult to get biomass out of there, all of which is entirely consistent with the notion that not all serpentinizing places are the same, that the chemistry matters a lot for the biology. And here's just another snapshot of the biology. So here are the fluids that we were able to sample from those wells. Here's the biology from the rocks themselves. And I don't want you to look at names or anything else. All I want you to see is that the colors here, each of which represents a certain genus of organisms, are very different than the colors here. So this is an entirely different community of microbes that we've accessed down here. And if we go back and employ this same methodology that I showed you before for trying to assess whether biology is at work here, whether metabolism is active, and the hydrogen and carbon monoxide in the system are being utilized, here's what we see. So again, here's hydrogen, here's carbon monoxide, this is a control, these are active. This is a control, these are active. And what we found is it's very, very difficult to see any significant evidence of biology at work. Even after 2 weeks of incubation here, 2 weeks of incubation here, using exactly the same methodology that we did before. Only in this one is there beginning to be some evidence of biology at work here. So this is significant, exciting for us for two reasons. The first is it suggests that we really have done something different by going down into the subsurface and getting away from the existing wells. We've sampled a different community, maybe one that hasn't had access to oxygen. And as well, we're starting to see the resolution of a gradient in pH. So higher pH is up here, lower pH down here, and there seems to be maybe a threshold. Maybe habitable here, maybe uninhabitable here. Sort of in a continuum, just as you predict from those models. So that's sort of where we are in this project at the moment. We have, as I've said, an active program of monitoring going on up there where we can go every few months and take another snapshot of the chemistry and the biology. And the notion is to sort of to make point observations that allow us to populate this theoretical plot of how hydrogen matters, how CO2 matters, how pH matters, and how all of it relates to the underlying geology. So just to briefly wrap up, I would say only a few things. The first is that habitability, including the habitability of these serpentinizing systems, is more than just binary. It's not either habitable or uninhabitable. Biology resolves itself across a spectrum of environmental conditions in many orders of magnitude of abundance. And that is a richness that we should seek to capture with our approach to habitability. And I think that we can. I think there are ways to do that. The diversity of serpentinizing systems in terms of chemical diversity and geological diversity is not very well captured by our sample set. And I think our notion of how these systems may be able to support life really is biased by the fact that that sample set doesn't capture the existing diversity. In fact, typically when people go to look for serpentinizing systems to study, they will look for the places with the highest hydrogen and the highest pH because they're interesting and exciting, and I would do the same thing. But it doesn't give you a complete picture of the biological potential in those systems. And finally, I would say that energy balance models of the type that I've tried to describe just now offer potential in a couple of regards. First, I think they offer potential to allow you to interpolate or extrapolate relative to observational data. So we have little point sources of data that we can do from looking at places like McLaughlin or other ophiolites, and it allows us to ground truth, our energy balance approach to habitability. And if we become confident in that approach, then it would allow you to take geological measures from someplace like Mars and make some notional predictions about what kind of biology that place might have supported or how abundantly it might have supported. And the other thing that I think it can do is because you can connect geology and geochemistry and biology all at the common language of chemistry, and more specifically at the common language of thermodynamics, it offers a potential to allow you to explore the relevant parameter space, the geochemistry, the hydrogen, the CO2, the pH, all of those things very quickly with models, right? Because we're all speaking the same language, the biology, the geochemistry, and the rocks. And that's what I'll leave you with. I will simply wrap up by saying that a number of different people supported this work. So as I mentioned, NASA Astrobiology Institute has supported it. The Sloan Foundation has a deep carbon observatory that has allowed us to do some monitoring work there. McLaughlin Natural Reserve and Barrick, which is the sort of reincarnation of Homestake Mining Corporation, all have been wonderful about providing access to McLaughlin and supporting that work. So thanks very much. Happy to take any questions.
Tori, I might start up with the questions. So you mentioned the failure of the carbon isotope in that particular sample that you were talking about because of the limitations on carbon availability there. Can you talk about possible implications for Mars there? Do you think that carbon isotopes would be relevant measurements that we can get to look for habitability on sites that we land at on Mars?
Yeah, so here's what I think the lesson is from a specific example of the use of isotopes to make inference about biology. The lesson, I think, is that you have to understand the factors that contribute to the production of an isotope effect and whether one should in fact be observed, and all the factors that need to be measured in order to give you a realistic view of what an isotope effect is. So the sort of shorthand way that people would typically do it is to say methane from methanogenesis is always light, meaning very, very depleted in C13, relatively enriched in CO2. And people kind of, they get used to seeing certain values of, minus 50, minus 60 or so, simply because you almost always have a carbon dioxide of about the same starting isotope. There's an ocean of it out there. So you kind of just have a notional baseline against which to measure. What really matters is the difference between the two, and it's important that you understand both. So the first lesson, I think, is that if you're going to make inference about biology on Mars or anywhere else, by looking at an end product and measuring its isotope, you have to have confidence about what the isotope value is of the starting material as well. Okay? So that's the first lesson. And the second lesson is, I think we have to be careful about the way we apply these things because there are times at which it doesn't work like we intuitively think it should. Almost everywhere that people look at methanogenesis, it is perfectly fine and perfectly viable to think of a relationship between biology and isotope effect. Because in almost no system besides these is carbon dioxide the most limiting thing. And only in this sort of unusual circumstance does the isotope effect fail to be expressed. But it shows you that environmental conditions can contribute, right? Isotope effect is just a potential that's there with biology, and you have to think about how the system actuates that potential or inhibits that potential. And those, I think, are the lessons.
Tori, let me mention either, depending upon your perspective, everybody's least favorite or most favorite MSL result, which is the non-detection of methane in the atmosphere. As everybody knows, you can make some methane inorganically, and we have seen evidence from spectroscopy that there are serpentine rocks on Mars. So were you surprised that there was no methane in the Martian atmosphere, and what does that mean?
From my understanding and what I have heard the atmospheric chemists say, I'm not terribly surprised that there hasn't been a methane detection yet, because it seems like there would have to be a very active methane source to give you something detectable. So I'm not terribly surprised, and I know the jury is still out, and I know they want to do some more measurements. But as you say, from what's been said so far, there's not a detection of methane yet.
There was an expectation from MoMA's work, of course.
Yes.
But had MoMA never published? I mean, just since we sent such a sensitive instrument to Mars, I think that without the sensational component of the whole methane story, we would have expected, I think, maybe to have seen a little. And it's interesting we see none at the limit of measure, and so that's what I was getting at.
Yeah, I think the most conservative statement that you could make about methane is that it probably is an indicator of water reacting with rocks in reasonable communication with the surface. So that's a blanket statement that you could make about serpentinization on Earth, is that almost always you see some methane associated with those systems. The jury really is still out on whether that is generally biological or whether it's geochemical. And when people look closely in laboratory experiments, there's not an obvious mechanism or an obvious observation of methane being produced abiotically. But it doesn't mean if you see it that it's biological. It simply would indicate, I think, neither of those things would be possible, neither the notional geochemical production of methane, nor the biological production of methane without water being in the picture somewhere.
Would we see the hydrogen that was produced by serpentinization with the instruments we have on the surface of Mars today?
That's a tough one. So I don't know whether SAM can see down to hydrogen or not. I don't know whether it can look at in a mass range that low. So I really don't know.
Thanks.
Question. Do you have any comments on habitability on Europa or Enceladus? And if not, what would you observe?
Yeah, so I would say, let's take the example of Europa and suppose that similar sorts of thoughts translate to Enceladus. I would say with respect to Europa, no problem with water, checking that box. Good arguments have been made that the biogenic elements, if those in fact are appropriate for thinking about life in such places, it would be abundant with no problem. For me, in those places, it really boils down to energy. And I would say, relative to the measure of what we know biology to require, we have uncertainty probably on the order of 10 to 12 orders of magnitude in how much biomass might be there if in fact there is enough energy at all to support it. So if I were going to make measurements to make the next important statement about the habitability of Europa, I would do a couple of things. So whether or not there are oxidants like peroxide, oxygen, whatever, in the surface ice that actually make it down will influence energy availability relative to a notional reduced ocean by an order of magnitude or two relative to no surface production of oxidants. So there's about 2 orders of magnitude there. Whether or not the ice turns over or how rapidly it turns over is another many, many orders of magnitude in terms of, even if you have a standing stock of oxidant in the surface ice, is it delivered and how quickly is it delivered? So I think there's a big difference there. I would try to make measurements that bear on that question. What are the concentrations of oxidants, if any, and how rapidly are they transported? The other thing that I think really would matter for Europa is it matters very much, I think, whether you have focused efflux of reductant into an overall oxidized ocean, as you would if you had hydrothermal venting, something like that, of hydrogen or sulfide-rich fluid into an ocean that is in general oxidizing because of delivery from the surface ice, versus a sort of comparable flux of oxidant from above, but in very diffuse fashion into an overall reducing ocean. I think those two would actually differ quite a bit, even though the theoretical delivery of energy would be about the same. The mode of delivery would bear significantly on the potential for biology. And I would say that the more focused case is the one that I would think of as having a more, I would be more optimistic about that. So in terms of actual measurables, anything that allows you to talk about the overturn of the ice would be priority number 1. Chemistry that allows you to say something about the oxidants, priority number 2. And then anything that allows you to model or constrain the potential for heat production and whether or not there may be hydrothermal circulation, that kind of thing at the water mantle boundary, to determine whether whether you might have some sort of focus venting. I would say that would be priority 3.
Yeah, my question deals with you're using molecular hydrogen consistently.
Mm-hmm.
What about simply the reduction potential of the assemblage? Is molecular hydrogen really necessary overall?
So I think it depends whether you take a geochemical or a biological frame of reference. If you take a biological frame of reference, so you're sort of speaking about redox potential versus actuated redox potential when you're speaking about geochemistry versus biology. So the biology needs to have something to eat, and there are maybe some indications that rocks can be directly dissolved and consumed, but from a more conservative perspective, the rocks need to react with the water on their own and deliver something like hydrogen or sulfide or whatever to the biology. So from a geochemical perspective, if you wanted to talk about redox potential in the system.
What about something on the surface, just surface chemistry? Chemistry on the surface of the mineral. In other words, the interfacial chemistry involving the organic phase on the one hand and the mineral surface on the other.
Yeah, so the thermodynamics for the biology get a little bit complicated when you start talking about surface attachment, because if it really is true surface attachment, so if there's no dissolution front that the microbe is sampling, but if it's actually accessing the solid phase, the way to do it would essentially be to create a wire between you and the surface and use it as an electrode. And such things, have been seen to exist. So there are polycytochrome wires by which organisms connect themselves to one another and to mineral surfaces and so forth. But thermodynamically speaking, the fact that that thing is a solid changes the way that you look at the energy yield versus a free molecule like hydrogen that can diffuse into the cell. You sort of just have to look at the solid as being an electrode with a certain redox potential. And that's not really the same for the organism as seeing the stuff come in.
Hi. So one of your the slides that was more interesting to me was the one where you showed outcroppings of crust from various geological ages on Earth,
Yep.
right? And Mars, because it doesn't have plate tectonics, and it's been a very long time since the Noachian age era on Mars. I'm wondering if that lack of movement of the rocks makes it, maybe less habitable or harder to study, or would have any kind of effect on the long term, because we're talking billions of years chemistry here, of any potential microbial inhabitant subsurface.
Yeah. So there certainly is a potential for these systems to be spent, right? So essentially, you could think of it this way: the overall potential of the hydrogen to deliver food to the microbes is a function of how much iron is in the rocks. And other things matter, like the activity of silica in the system, all these sorts of things. But let's say, to first approximation, for every iron that's in the rock, you can make half a hydrogen, basically, if you turn the reduced iron into magnetite. So you could say, a kilogram of rock has a certain potential where hydrogen and where biology is concerned. In order to actuate that potential, you would need the water to be able to access every portion of the rock. So a couple of things might happen. You could have local movement that fractures the rock and creates new surfaces. The process of serpentinization itself has a large partial molar volume change associated with it. So there is conceivably some self-fracturing that goes on, but not necessarily enough to make every part of the rock accessible. And that's significant because this is a process that doesn't just make end products that diffuse away. It makes a variety of mineral end products. And if you happen to plate those out on the surface that you're working on to begin with, you inactivate that surface. And so there's a lot going on to sort of look at how does that actually work. If you have a certain number of grams of rock in the system and you allow it to react, does it matter how finely divided it is? Or can the water ultimately access all of the rock in the system? So in those terms, I would say, yes, it matters whether or not there is activity that can expose fresh surface. It certainly also matters whether or not fresh material is being brought up at all. And from the perspective of supporting biology over the long term, although simply to support biology, what you would want is to have the process go fast to deliver lots of reductant. To support biology over the long term, you'd want some kind of optimum in the middle, right? You'd want enough to keep the biology going, but also to not spend your entire supply, in the first little while. So, as a quick answer, I would say yes, it probably matters to think about the age of the crust. And part of this plot that you see here, and part of the project that Dawn, this postdoc who worked on this, was doing, was to look at ophiolites of differing ages to see whether or not you find yourself at different points in the sort of geochemical evolution and therefore in the biological evolution of the system.
Okay. Back to the isotope chemistry of methane. My daughter's current postdoctoral research is on methane emission from natural systems. And her instrument, a laser instrument, has recently been upgraded to detect the difference between hydrogen and deuterium in the methanes, and that has a relevance for the source of the methane.
It does.
Could that give you a new handle where the carbon thing didn't?
Yeah, potentially yes, but HD is more complicated than C12, C13 for this reason. In C12, C13 systematics, when the CO2 comes into the cell and is turned into a methane, you could track it. If you painted it red, right? If you painted the CO2 red, the methane that comes out is also red. In the case of the HD on the methane, it is not the case that the hydrogen that comes into the system is the hydrogen that leaves on the methane. So what happens is hydrogen, so if, in other words, if you painted the hydrogen red, the methane would largely come out uncolored. And the reason is hydrogen comes into the system, enzymes break it apart into electrons, essentially, that become part of electron transport in the cell, and protons. And the protons become a part of the overall pool of protons in the cell. So essentially, the instant the hydrogen comes in the cell and is tackled by the enzymes, that H, or that D, becomes part of the overall pool of water. And you still can wind up with an isotope signature in methane in a couple of ways. So if the methane is made from acetate, which may be what your daughter is working on, then you actually can follow the hydrogens, because they come in stuck to a carbon and they leave stuck to the same carbon. It's that carbon that becomes the methane. If we're talking about CO2, and in a place like Mars, for example, the potential to get acetate as a precursor is much lower. Then you have the more complicated interpretation of how is it that methane produced from hydrogen and CO2 acquires an isotope signature? How is it that HD ratio changes? It does because as metabolism happens faster and faster in the cell, water exchanges in and out of the cell. Hydrogen is produced during the course of metabolism within the cell, and the relative contribution of those two makes the intracellular water a little bit different than the extracellular water. So the faster it goes, the more that signature is imposed. But that's a much more complicated argument to try to make when interpreting the signature of methane in the system.
Just two more questions.
Yeah.
Okay, I guess my old geochemistry days, is this working? Okay.
Yeah.
Is there a poisoning effect from things like trace elements in serpentine like nickel that might affect what you observe in the analogs, that in addition to your hydrogen and your everything else you're looking for that as suitability for life, that would actually prevent or kill a life? It certainly does so for the plant choice.
Yeah.
And I just didn't know if that's happening deep underground as well.
Yeah, potentially, yes. So ironically, actually, nickel is great for methanogens. The main enzyme that they use requires nickel, and so that particular one is good. But I mean, if you started talking about chrome or things like that, yeah, I mean, potentially, yes, and I think you'd have to dig into the biochemistry a little bit to understand whether you are, competing for enzyme binding or something that really would represent a toxic effect. Short, honest answer, I don't know without trying to dig into the biochemistry a little bit more. But it's an important point to make as another example of the fact that not all serpentinization is the same, right? They're even at the level of trace chemistry, it might matter.
I'm wondering if any people studying serpentinization regard that as a possible environment for the origin of life, since you can't really have something inhabited, even if it's inhabitable.
Right. So I'll say two. Parameter of choice for habitability was biomass density potential, if you wanted to think of it that way. Simply because it's something that's easy to quantify, it's easy to measure in the world around us, and it's something that can be predicted from this modeling. You could equally well choose any one of a number of different parameters, one of which would be the potential for life to evolve, right? Because it is, as you say, a precursor for life. I would say we understand so little about how that process works that although it's a viable parameter, a valid parameter, it's not one that we actually could use or quantify. But it is something that people are interested in. And Mike Russell, who I think gave a talk here probably a few months ago.
April, yes.
So Mike Russell has been a very big advocate of life arising in serpentinizing systems. And it's partly because, to sort of quickly summarize the way that Mike thinks of it, you could say that life has two basic problems to solve at the level of metabolism. One is to get energy, and the other is to turn CO2 into sugar or other reduced carbon. So you need a source of reductant and you need a source of energy. And Mike's notion is that all of the hydrogen, so the very reducing character of these fluids, gives you that reductant that you need. And the fact that it's very alkaline, taken in reference to an ocean that's less alkaline, gives you an energetic potential. So life uses gradients in protons usually, or occasionally in sodium, to store energy in the form of chemical and pH gradients. And Mike's notion is, if you have a fluid that's coming out at pH 11 or 12 into an ocean that's at pH 7 or 6 or so, there's energy stored in there. And he sort of goes into the details about it more. But yes, long story short, there are people who have thought about it in that way, and Mike is the one who does it most.
And that's online, right?
That's online. If you're watching this online, I suggest you watch that one next. So Tori, we have a special mug here, SETI.org/talks mug, where there's a couple of androids there talking about how likely it is for life to be elsewhere, and tackling the question of planetary habitability is what we're all about here.
Great.
So please join me in thanking Tori for his great talk.
Okay. Thank you.
Further questions, please feel free to come up and chat to Tori now. Well, actually, yeah, be quick with them, actually, I think. You have